Okay, so this is why science is so cool. So right now I'm doing it's an H258, which is HOX, which stands for nucleated cells. So you can see like this density region right here. So this is all thymus. So this density region right here, when they're more packed together, that's considered the cortex. And this loosely packed region is supposed to be the medulla. So, and you can do it this way and you can see like, okay, the more packed, so we can assume that we would outline this as the entire cortex and maybe some of this down here. But it's something really sick. So now we're gonna be doing our other antibody, which was binding to CD86, which is a marker for stromal cells kind of thing. It really marks the medulla of the thymus. So what we should see is when we turn on this wavelength of light onto our confocal, we should then see green only really light up this region, but leave this region kind of the way it is, like dark. So now we're going to stop our process right here, which was a light imaging. We're gonna finish with that, and now we're gonna use our 488, which is like green wavelength. Click live, and boom, you can see right here, this was what we predicted. We don't really have the dark green here because there's densely packed cells, so there was low levels of CD86, but then here, boom, a ton of CD86, confirming that really it was the medulla that we were referring to. And it's pretty cool. We can go here again, and the last one I have is a 546. So what this one did is it's going to be telling us where all the dead cells were. So now when I click live, you can, so you can see like it's only one dead cell here, maybe a small one right there, right here, and we can adjust the tuning to try and get the maximal amount. So you can see like, that's probably the best we're gonna do. Right there probably is the best. You see some tiny marks up here. But you have to run it through another program to see if those are actually true marks, but these for sure definitely gonna be marks. And you can see kind of like where in the cell the cleaved caspase 3 was marked. So it's really, really interesting. So yeah, so if we want to like focus on this area right here of the cell, we can stop this. Watch out, it's gonna be blue right now, so be careful. So there is a big absence in that area because the DAPI was not able to bind to the nucleus because it was already destroyed by the cleaved caspase signals. So that's pretty much a introduction into confocal microscopy.